Hello, dear students. Today we are going to discuss on DNA replication part 1. DNA is considered as the central molecule of living system because it acts as a memory bank of genetic information. All of you know. Genetic information flows from DNA to RNA and then to proteins. The DNA is duplicated by the process called replication and transferred to the daughter cells during cell division. The copying of DNA must occur with utmost accuracy because any error may result in deleterious consequences. So let us discuss the three processes like replication, recombination, and repair and their role in maintaining the integrity of DNA molecules. So today we'll discuss on the replication and we'll focus on replication, and this is the class for replication part 1. So principles of replications includes the following. Number 1, copying in accurate. So as the carrier of genetic information from generation to generations, the integrity of DNA must be preserved at any cost. Okay, this needs accurate replication. Any error in replication not only compromises the function of the gene, but is also heritable. Taking into consideration the enormous size of human genome, that is 6 × 10⁹ base pair spanning 23 pairs of chromosomes, which is an accurate replication that is not a very easy task. To overcome the problem and to achieve fidelity, the process of replication is endowed with proofreading activity. This is very important. So the next one is replication is semi-conservative. Replication is the process of doubling of DNA that occurs by copying of the DNA sequences by complementary base pairing. That is A with T and G with C. The double helix of DNA is separated into 2 single strands and is replicated with each strand acting as a template for a new strand. In this way, half of the parental DNA molecule, there is one strand, is conserved in each of the newly synthesized daughter DNA molecules. In this way, each of the newly synthesized DNA molecules contains one new strand and one old strand, and hence the replication is considered to be semi-conservative. This is number 2. Now, number 3, supercoils and DNA topoisomerases. DNA bends on itself and forms supercoils that tightly pack the DNA into the compact structure. This imparts a high degree of structural stability into DNA, but the 2 vital processes like replication and transcription need free access to the information into otherwise tightly packed DNA. In other words, both processes require the acing of supercoils and separation of DNA strands, which is not a simple process. When an attempt to separate the strands, the DNA double helix is unwound, which leads to successful overwinding or winding in surrounding regions. And a group of enzymes known as DNA topoisomerases solve this problem of supercoiling. And this enzyme cut the DNA strands and release the strain caused by overwinding and then reseal them to overcome the problem of supercoils. Okay. Now, the next thing is the replication fork. The question may arise. So this is the replication of DNA. So there are different principles of replication, what I am explaining. So fourth one is the replication fork. The question may arise here are the parental DNA strands unwound completely before replication. The two parental strands are separated simultaneously into the localized region that results in the formation of replication fork. The separated strands are immediately replicated. The separated strands are immediately replicated. I'll show you this picture. Yes. The separated strands are immediately replicated and the replication fork moves forward along with parent DNA. Enzymes known as DNA helicases, which act as zip openers, are responsible for the separation of strands as they move along the DNA helix during replication. Helicases utilize ATP for the energy supply during this process. The replication always starts at a unique point, which is called origin of replication, which is a single in prokaryotes and double or multiple in eukaryotes. It's better to say multiple in eukaryotes. And these sites are usually short sequence of AT base pairs. Now, number 5. What is that? Replication is simultaneous and bidirectional. Replication of DNA occurs in the 5' to 3' direction. Okay? Simultaneously on both strands. The statement may appear self-contradictory because when the synthesis occurs always in 5' to 3' direction, how can both strands be synthesized simultaneously? Remember, both strands run anti-parallel to each other. If both strands are synthesized continuously while the replication fork moves on, one strand would have to proceed in 3' to 5' direction. This paradox was answered by Okazaki in 1960. They showed that one strand, which is called leading strand, is synthesized continuously, whereas the opposite strand, that is the lagging strand, is synthesized in small pieces, which is called Okazaki fragments. In a discontinuous manner, the size of Okazaki fragments range between few hundreds to few thousands of nucleotides in different cells. The Okazaki pieces are ligated together later to form a continuous strand of DNA. Okay, leading strand proceeds 5' to 3' in the same direction as that of replication fork movement, whereas the lagging strand runs 5' to 3' opposite to the direction to the replication fork movement. Okay. Now the enzyme in number 6. What was the point in number 6? So this is DNA synthesis is catalyzed by DNA polymerases. So the enzyme that synthesizes DNA is known as DNA polymerase or DNAPs. The deoxyribonucleotide triphosphate, that is DATP, DGTP, DCTP, DTTP, etc., are added by DNAP, that is DNA polymerases, one after another. as per the complementary base pairing on parent strand to the 3' end of the growing strand. Okay. So DNAP DNAPs cannot initiate replication as such. They can only add nucleotides to the pre-existing strand, which is called primer. So primers are short fragments, which is having 5 to 50 nucleotides of RNA rather than DNA. Primers are synthesized by RNA polymerases. In addition to the main function of nucleotide polymerization or nucleotide polymerization, DNA polymerases also have the intrinsic proofreading ability. So DNA polymerase double checks each nucleotide after it is added to the incorrect bases incorrect bases are removed before the phosphodiester bond is formed. And this is how a high degree of fidelity is maintained during replication. So let us know about the sequence of events of DNA replication in prokaryotes and eukaryotes. So this is something I have written on discovery of Okazaki fragment. So this you can find in any book. So replication in prokaryotes. The process of replication can be studied conveniently in 3 stages. Okay. What are the stages? Initiation, elongation, and termination. So first we'll discuss on initiation. A specific protein named DNA-A locates the site of origin on parental strand, which is AT rich region. Helicase enzyme bind to the DNA strands and start unwinding them. And this leads to formation of a bubble-like structure initially that eventually forms the replication fork. Okay, I have written here replication fork. Helicase enzyme is the initiator. Now, separation of strands by helicases produces stress in supercoiled structure of DNA, and which is relieved by topoisomerases, and which is called as DNA gyrases. So DNA gyrase and topoisomerase both are same. Further, a group of protein which is called single-stranded DNA binding protein, which is in abbreviation SSB, single-stranded DNA binding proteins bind to single-stranded DNA separated by helicases and prevent them to form the recoiling until the replication is completed in that region, right? The synthesis of the new strand of DNA does not start as such. It needs a pre-existing primer, what I have discussed earlier. The primer which is present on the template strand is short segment of RNA polymerase. Okay. So it is interesting to know that the single primer occurs on the leading strand, whereas the multiple primers do exist on the lagging strand of DNA. At this stage, replication is initiated by enzyme DNA polymerase 3, that also I have mentioned here, and which adds a deoxyribonucleotide residue to the RNA primer molecule. So this is about initiation. So this is the diagrammatic representation, and next thing is elongation. Okay. So the synthesis of new DNA strand by DNAP3, that is DNA polymerase 3, occur antiparallel to the template DNA strand as per the complementary base pairing, that is A with T and G with C, and proceeds in 5' to 3' direction. The incoming deoxyribonucleotide residue are added one after another to the 3' end of the growing DNA strand. Right? Leading strand synthesis begins with the RNA primer at the origin of replication site. With the sequential addition of deoxyribonucleotides by DNAP3 or DNA polymerase 3. Leading strand synthesis proceeds continuously, uninterrupted, as a parent DNA is separated by helicases. So lagging strand synthesis is not so straightforward. So DNA synthesis on the lagging strand poses some problems, since DNA polymerase enzyme cannot add the incoming nucleotide to 5' end of the growing chain. DNA synthesis on the lagging strand occur as small pieces, and these fragments are synthesized discontinuously on the lagging strand, and they are called Okazaki fragments. They are called Okazaki fragments. Okay, and once the synthesis of an Okazaki fragment has been completed, replication stops with DNAP3, which dissociates and reassociates with a new RNA primer to synthesize a new Okazaki fragment. Right? Next, once an Okazaki fragment is fully synthesized, its RNA primer is removed and replaced with DNA fragment by another enzyme which is called DNA polymerase 1. The gap between DNA synthesized by DNAP3 and a small fragment of DNA synthesized by DNAP1 is bridged by the phosphodiester linkage. And this Nick sealing reaction is catalyzed by an enzyme called DNA ligase, and which utilizes ATP for this purpose. Apart from the main function of DNA synthesis, DNAP3 also possesses proofreading ability, and it checks the incoming nucleotide and allows only the complementary base pairing, preventing any sort of mismatching, and this ensures a high degree of fidelity during replication process. Okay, and the last part is termination. What about the termination? The synthesis of DNA continues until it reaches a region known as terminus sequence. Once the replication fork enters the terminal region, it cannot make an exit, and replication comes to an end. So today's discussion includes the replication of prokaryotes with the basic principles of replication and Okazaki fragment we have discussed here. And now I'll show you one animation on this topic. Just follow this one.
Replication begins with a sequence of nucleotides called the origin of replication. Helicase unwinds single-stranded DNA helix, and a single-strand binding protein reacts with a single-stranded region of the DNA and DNA polymerase 3 is the major enzyme involved in DNA replication. DNA polymerase 3 can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase called a fragment constructs an RNA primer sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase 3 can then add deoxyribonucleotide to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are anti-parallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase 3 reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase 1, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further along new prime and DNA polymerase 3 jumps ahead, begins synthesizing another Okazaki fragment. For simplicity, DNA polymerase 3 has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase 3 is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase 3 are not shown.
Dear students, I hope all of you have enjoyed this animated video and wish you all the best. Just go to your textbook and find what else are there. Hope to meet you again on your next class on DNA replication part 2. Thank you.